The objective of this practical is determination of red blood cells per cubic millimeter of human blood. Now we will understand the principle of this practical. Red blood cells or erythrocytes are round, anucleated biconcave cells which contain very important protein called hemoglobin. This hemoglobin is responsible for transport of oxygen from lungs to different tissues of the body. These red blood cells have a lifespan of 120 days in the circulation. As red blood cells become fragile, they present membrane surface factors that are recognized by splenic macrophages which destroy the old red blood cells. The red blood cells count along with other tests provides valuable information to the physician in diagnosis of different types of anemia in patients. What are the requirements for this practical? In order to perform this practical, you will need a hemocytometer, a microscope, blood lancet, alcohol or spirit swabs, RBC pipette and Hems fluid. This is how an RBC pipette looks. It has a red bead present inside the bulb. This RBC pipette has three markings. First is 0.5, which is present in the middle of the stem. Second is 1, which is present at the junction between stem and bulb. And third is 101, which is present above the bulb. This is the composition of Hems fluid. It contains sodium chloride for isotonicity, sodium sulfate which is anticoagulant, mercuric chloride which is antifungal and antibacterial, and distilled water. What is the procedure of this practical? First, get a finger pricked under aseptic conditions. Wipe the first 2 or 3 drops of blood and let a good sized drop of free flowing blood to form. Then place the tip of the clean and dry RBC pipette at the edge of the blood drop. Then suck the mouthpiece of RBC pipette and draw the blood up to the mark 0.5. Then suck Hems fluid up to the mark 101 and then mix the contents of bulb for about 3 minutes. Then discard the first 2 drops of fluid from the pipette and charge the hemocytometer with the diluted blood. Ensure that there is no blood overflow into the trenches and there is no air bubble under the cover slip. Then allow the cells to settle down for about 3 to 4 minutes. Then place the chamber under the microscope. Use the LP lens to focus the grid lines and to check the even distribution of the cells. Choose a combination of condenser and diaphragm for optimal illumination. The red blood cells are to be counted in 5 squares, 4 corners and 1 central group of squares each, that is, in a total of 80 smallest squares. Then switch to HP lens and start counting in the left upper groups. Count the red blood cells in each smallest square, including those lying on its left and lower lines and ignoring those lying on its right and upper lines, because these cells will be counted in the adjacent squares. In this way, you will avoid counting the cells twice. While counting the cells, continuously rack the fine adjustment screws up and down so that the cells sticking to the underside of the cover slip are not missed. What is the dilution factor? Note that the stem of RBC pipette only contains the diluent while the bulb contains the diluted blood. So, the dilution of the blood occurs in the bulb of RBC pipette. Since the volume of the bulb is 100, that is 101 minus 1 equals 100. When the blood is taken to the mark 0.5 followed by diluent, that is Hayem's fluid, to the mark 101, then the volume of blood is 100. It means that 100 volume of diluted blood contains 0.5 volume of blood and 99.5 volume of diluent. This gives a dilution of 0.5 in 100 or 1 in 200 or 200 times, the dilution factor being 200. If blood is taken to the mark 1 and diluent to the mark 101, then the dilution obtained would be 1 in 100 or 100 times, the dilution factor being 100. Now we will understand calculations for counting red blood cells. The red blood cells are counted in 5 groups, that is 4 corner groups and 1 central group, each of which is divided into 16 smallest squares. Therefore, the red blood cells are counted in 80 smallest squares. Each side of one smallest square is 1 by 20 millimeter. So, the area of each smallest square will be 1 by 400 millimeter square. The depth of each smallest square is 1 by 10 millimeter. Therefore, the volume of one smallest square will be 1 by 4000 millimeter cube. So, the total volume of 80 smallest squares will be 1 by 50 millimeter cube. Let the cell count in the volume 1 by 50 millimeter cube of diluted blood be X. Then 1 millimeter cube of diluted blood will contain this amount of red blood cells. When we multiply this amount with the dilution factor 200, we will get the number of red blood cells present in undiluted blood. After performing these calculations, we will get this result. It means by multiplying the number of red blood cells counted in the 4 corner groups and 1 central group with 10,000, you will get the total red blood cells count. Now we will understand counting of red blood cells. This is how red blood cells appear on a Neubauer's chamber when this chamber is placed under a microscope. The red blood cells are counted in a similar way as white blood cells. The only difference is that these red blood cells are counted in 5 groups, that is 4 corner groups and 1 central group, each of which is divided into 16 smallest squares. The amount of red blood cells counted in all these 5 groups is then added. This amount is then multiplied by 10,000 to get the total red blood cells count. What are the precautions? Observe all precautions for getting a finger pricked, filling the RBC pipette and charging the hemocytometer with the diluted blood. Clean the hemocytometer with a lint-free piece of cloth. Once cleaned, do not touch its central part because oils from your skin might affect cell counting. The cover slip should also be grease-free. Continuously rack the fine adjustment screws while counting the cells for accurate counting of cells. Here is your study question. What are the other techniques for counting red blood cells?